All right, moving along then. That was phase 1 of the cell cycle, interphase. And remember, interphase has 3 parts: gap 1, synthesis, and gap 2. The second part of the cell cycle is actual cell division, and that's where we're going to split those sister chromatids apart from each other. There's 2 parts to this. There were 3 parts to interphase, but there's 2 parts to actual cell division. First, we have to divide the nuclear material, so in other words, the sister chromatids, right? The DNA has to be divided equally between the 2 cells, so they both have the same sets of instruction. And this is what we call mitosis. We'll be talking a lot about this in the next few days. Okay, so mitosis is dividing up the DNA. And then the next part of cell division is called cytokinesis. That's where the cytoplasm divides. So all the stuff that's in the cytoplasm, the ribosomes, the mitochondria, if it's a plant cell, the chloroplasts, they have to get divided equally into the different cells as well. Cyto refers to the cell. Okay? Kinesis means to cut. So this literally means we're going to cut the cell in half. So once we've got the nuclear material, the DNA, divided up, then we're going to cut the cell in half so that all the stuff in the cytoplasm can also be divided up. So, to look at this in one big diagram, which helps me to see it all kind of together visually, we're going to go through this little pie chart kind of thing, and it may help you to draw this out in your notebook as well. Remember, we had 2 parts to the cell cycle. Okay, it shows those as 2 pieces or 2 parts of the pie. This whole section here is the first part we talked about, interphase. Where I said interphase takes up the majority of a cell's life. Gap 1, growth and development. S, or synthesis, that's when we make those copies, those sister chromatids. And gap 2, when we make the centrioles, which we still haven't talked about yet, and we'll get to that, what those are. Okay, so that takes up the majority of the time of the cell's life. But when the cell is all grown up and ready to make its own cells, and it has all the chromosomes copied and all the organelles made and everything else, then it's going to go into mitosis. That's this little slice of the pie. Remember, mitosis is when we divide up the DNA. As you can see, that takes a very short period of time. And then, after it's done with that, then we can divide up the cytoplasm. There's cytokinesis. Also takes up a short period of time. Okay, both of these things are part of cell division. Okay, or the mitotic phase, it's sometimes called. Okay, but this is where the cell actually splits. You can see, hopefully, that we start with one cell right here. It goes through all of these stages, interphase, grows, copies its DNA, makes centrioles, gets to mitosis and cytokinesis, and pops out a new cell. And then both of these cells now are going to do the same thing again. They're going to go through gap 1, synthesis, gap 2, all parts of interphase, and then they make copies of themselves through mitosis and cytokinesis and pop out a new cell each. And so that's why I said when we are developing as an embryo, we can go from 1 cell to 2 cells to 4 cells to 8 cells, because all of those cells are going through this cell cycle. All right, one of the ways that you're going to practice the stages of mitosis, so that actual nuclear division, is by doing some modeling. And the mitosis modeling is going to end up being graded as a quiz, kind of like the microscope quiz that you wrote back when we started studying cells, and the quiz was either an all or nothing thing. You just kept taking the quiz until you got it right. Same thing's going to happen with this, but instead of a written paper and pencil quiz, as I said, this is going to be a modeling quiz. You are going to show mitosis, or in other words, how the cell divides its DNA with actual materials that represent the different parts of the cell. And while you show it, you're going to discuss what's going on in the cell using some keywords. So first we're going to talk about the materials that you're going to be using. You're going to need a large string that's tied up in a circle that represents your cell membrane. You're going to need a small string tied in a circle that represents the nuclear membrane, so the membrane of the nucleus. You're going to use some beads that represent the chromatin to begin with, which turns into the chromosomes, which then turns into sister chromatids, and then I'll guide you through this to tell you how this is all going to work. And then you're going to use some tiles, some little mosaic tiles that represent centrioles. The first stage you're going to start with is called prophase. Remember, these are parts of mitosis. You do want to write this down in your notebook so you have this as a reference while you practice your modeling. So what you want to do first is you're going to describe your cell. So I'm going to show you how you set up your cell here. So here's your large string that represents the cell membrane. Here's your smaller string, which represents your nuclear membrane. Okay, so I'm starting to describe my cell. Here's the cell membrane, here's the nuclear membrane. Inside the cell, we have different chromosomes. Now, the beads that you're going to be using for these chromosomes come in different colors. So I'm going to use some red chromosomes and some blue and some yellow. Okay, there's my chromosomes inside my nucleus. Remember, the chromosomes are made up of the DNA. We know the DNA is in the nucleus because that's what gives instructions to the rest of the cell. We also have some centrioles. Okay, the centrioles, as I said, are little mosaic tiles. They also come in different colors, but I think I'll just make them black. So in prophase, we've got a cell membrane, we've got a nuclear membrane with chromosomes inside, and there are two centrioles. These are those little mosaic tiles that are in the cell as well. Again, you don't know what these centrioles do yet. We'll get to that in a minute. So what happens in prophase? In prophase, and you may want to write this down for yourself as a note, the chromosomes become visible. Remember, they already have made copies of themselves as sister chromatids. Okay, so they're already doubled. So you're going to have two beads of each color, but they're just kind of like piled up in the nucleus, not really in those stick-like shapes before prophase. But once prophase starts, then they kind of become more stick-like so you can see individual chromosomes. Okay, the nuclear membrane begins to disappear. Okay, and the centrioles move to opposite sides of the cell, just like I have here, centriole here and a centriole here. Okay, so they move to the opposite sides, so they're all set up. Okay, that's what happens in prophase. Again, your chromosomes start to become visible, so you'll show that with your beads. Okay, your pairs of sister chromatids are now visible. The nuclear membrane disappears, which you'll show by just removing this string here. Okay, and then the centrioles go to opposite sides of the cell, which you'll show by just moving them to opposite sides of your cell membrane.